Welcome to the NLM Informatics lecture. Our speaker for today is Dr. John Gennari from the University of Washington. Dr. Gennari has been a professor in biomedical and health informatics for over 15 years. His doctorate and background is in computer science, but he's been working in biomedical informatics since the early 90s, beginning with work on the Protégé knowledge representation ontology development system. He has a wide range of experience on large multi-institutional, multi-disciplinary projects, and this led to his research focus of knowledge reuse and knowledge sharing. His expertise is in ontology, development, standards, and semantic web tools. Dr. Gennari is the graduate program director at University of Washington, overseeing the PhD and master's in the research programs. He is currently on the NLM Biomedical Informatics Library and Data Science Review Committee. Dr. Gennari.
Okay, check. I've got a lot of lead here, so I'm going to wander around. It's going to be my style. What did I do with the little? Here we go. All right, thanks for the introduction, and thanks for coming. Yeah, so that's the title of my talk, and I'm just going to jump right into sort of an outline or some bullet points I want you to take away from this talk. And as you gather from my introduction, I'm interested in semantics, or how you represent things. And in particular, this whole talk will focus on the semantics of biological processes. More specifically, the bulk of the talk is going to be about model merging, and I'll explain that in great detail. And then at the end of the talk, I've got kind of two more forward-looking, more longer-term endeavors that we're working on, having to do with building infrastructure. It's kind of unglamorous plumbing work, but it's really important. And reproducibility, which is a hot new buzzword, and we've got some new developments there to tell you about. Just to color things up a little bit, these are some of the logos that we'll be using through the talk. The OPB stands for the Ontology of Physics for Biology, which is exactly how you encode the semantics. And SemGen is the tool for model merging of biosimulation models that I'll talk about in great detail. And at the end of the talk, I'll talk a little bit about a brand new Center for Reproducible Biomedical Modeling that is currently being funded with the start date, I think, of June 1, 5 days ago. So I can't tell you much about it, but it's very exciting. Okay, so let's start with biological processes. I'm not very familiar with the audience here, so I'm going to sort of start from ground zero in a lot of ways. And I do know that this is where reporting on funding from the National Library of Medicine that I've received the last 4 years. And I know from being on the study section that my work is a little bit unusual. So actually, I think it's useful to start at ground zero on this kind of thing because, yeah. So what's a biological process? I'm going to use the word biosimulation models. And frustratingly to me, that's not a word you'll find in the literature. If you do a search on that, you won't see too many papers about it except for my groups. People tend to just use the word model, and I hate the word model, not because it's a bad word, but because it means so much. So many different things can be models. So I'm going to use the word biosimulation model in particular. And then we'll talk about, and you'll see why it's hard to represent these things. So kind of the right thing to do here would have been for me to show you a whole bunch of clips from YouTube. Because if you're talking about a biological process, you're talking about something that unfolds over time. That's a key component of what a process is. But I was too lazy, or the technology was too complicated, or seemed too glitzy or something. So I'm just going to ask you to imagine these kinds of things happening over time. And in fact, if you do a YouTube search for any of these phrases, you'll find a billion, especially like muscle contraction or actin and myosin. Oh my God, there must be like 20 YouTube videos of little animations of the muscles of the actins and myosins shrinking together. But we can have a wide range of things that might be included under biological processes. One of the goals of my group is to be able to represent any and all of these. So blood flow through the heart is obviously kind of at the gross organ level. Muscle tissue contraction would be at the tissue level. But then the rest are kind of at the cellular level or even subcellular. I'll actually talk a little bit about calcium ions flowing through a channel in the membrane, in a membrane. So here are some of the aspects you might think about when you're thinking about these kinds of processes. First of all, there are multiple scales of time and space. I already referred to that, right? So things can be really big or really small. And all of them have a temporal aspect. And a key part of a biological process and a biosimulation model is to be able to say exactly how fast something is happening so that you can synchronize things and figure out when it's going to produce. And then the other thing that's on these examples is some of them are for physiology and some of them are pathology. I guess cancer is the only one there. But usually you're interested in these models of physiology if you want to know what happens when it breaks, right? So if you're interested in diabetes, you're very interested in beta cell insulin production. If you're interested in hypertension, you might be interested in, well, any number of things. And if you're interested in cancer, you might be interested in tumor cell mitosis. Okay, so here's my definition of a biosimulation model. So you have a process that occurs in reality, and what you're going to do is you're going to abstract that and make a simplification of it that is a mathematical abstraction. Then, to really be a model, it's not just a set of equations, but it's actually some piece of code or something that runs, something that you can use to simulate the process over time. Okay? So it needs to produce, you need to be able to say go, run, execute, to produce some temporal trajectories of something, of properties, physical properties that you might be able to measure, like blood pressures or like calcium concentration. So here's what a trajectory might look like. And this is, in fact, calcium concentration and muscle contraction. And so the calcium concentration is leading to increases in force. And I'll just zoom in there and you can see that picture. The key thing is the x-axis is time. That's the unifying thing across all biosimulation models, is that you're, at the end of the day, you're producing a temporal trajectory. And you're going to do something. Well, so what? I mean, that's the question. What are you going to do with that? And there's lots of things lots of people might do about it. I mean, first of all, it's finding out about biology. So if you know more about biology, you might actually be able to figure out some kind of drug design, right, to disrupt that. It also might be patient-specific modeling, right? So if you can parameterize a particular mathematical model with an individual's data, then you might be able to predict how that individual is going to, what might happen to that patient over time by running a biosimulation model in advance. Oh, wait, yeah. So I guess the point of my next set of slides is that, those are some ideas about what you would do with them. But in truth, sort of the killer app for biosimulation modeling really isn't out there yet. Okay? One of the killer apps for the internet was Google Maps, right? Suddenly you could navigate, which you couldn't before. I mean, I'm old enough to remember this. The really younger generations, of course I know where I am. But at any rate, that was a big killer app for the internet, was that suddenly you could navigate. So what are the killer apps for physiology? Well, let's just think about this in the big picture. So physiology is complicated. There's lots of things going on. Everything is connected to everything. One thing leads to another. But we know that if you eat too much cookies and soda, you'll get fat, maybe. Or that seems to be a plausible process, right? The question is, just like on Google Maps, you want to know how it happens. How do you get there? How fast? How long does it take to get there? And those are questions that you might answer with physiological modeling. Okay, so you eat cookies, that breaks down into glucose, and then what happens, right? This is the kind of question you might ask. And at a crude level, or slightly more detailed level, so the glucose goes into the blood, and then enters the fat cells, and then insulin occurs, and things go up and down, and eventually obesity goes up. That's kind of the idea. This is very crude, of course. And to really make this a mathematical model, you have to have rates, and you have to break that steps down into individual steps at whatever level of abstraction you're working at. So that's kind of the idea. And this is the idea of the physiome, right? So the genome is all the genes, the proteome is all the proteins. The physiome wouldn't it be great if we could catalog all the physiological processes that are happening in organisms. Okay, and this term and this picture is courtesy of Peter Hunter, his publication in 2003. And the point is, as I've already hinted at before, we have to think about multiple scales of both space and time. And because it's so complicated, because so many things touch each other, so many things interact, we have to have reusable components. Okay, so to me, this is one of the driving forces behind the notion of reuse and plug-and-play modules. If we have reusable components that are plug-and-play, we can build better models. And as I alluded to, that leads simply to better scientific understanding of the pathophysiology that then we can target with drugs or whatnot. And it might lead to patient-specific predictive models of disease. But those are kind of out there still. Okay? Okay, so to build this physiome, what I say we need is Legos. Okay? So those are the prototypical, everybody knows exactly how Legos are plug and play, right? This is a great analogy to use. They're standardized, right? That's the whole beauty of it. Those nibs are exactly the same. They fit into each other. And so then you can take them apart and put them together as many times as you want. That's what we need for these models. So I love this analogy both because everybody gets it right away, the whole notion of plug and play, and because I actually do like Legos. So ultimately, we need a lot of Legos. And this is a set I built with my daughter. So that's what we'll eventually need. Okay? So that's part one of my talk. Now, kind of the longer part of my talk, I should have a clock where I can see. Oh, good. Is the details of some of this plug and playing, right? So we want to put these Lego pieces together. How are we going to do that? And that's what we're going to explore next. Okay. So the first problem is to find the Lego pieces, right? How do researchers find models? Maybe they do a literature review. Maybe they do a GitHub search. If they're actually looking for code, right? You'd have to do a GitHub search. And can they understand what they find? And then can you actually plug them together like Legos? That's the whole idea. And we have some experience in doing this kind of thing. I've been, as part of this grant, I've worked closely with 2 repositories where people put their code for biosimulation models. The CellML repository, which Peter Hunter is involved with, and EBI's biomodel database. But what I'm going to say is that although these are a good start, they're just a start. There's lots of problems with them. And as I said, we've worked closely with these groups, so these groups are actually the first people to tell us about the problems. And I'm not making them up absent of knowledge. So again, starting from square one, I'm going to tell a simple story about this kind of model merging that I envision. Okay, so Jane has an idea. Maybe she's attending a talk like this by Derone. I hope there's nobody whose real name is Derone. I totally made this up. And she has a model of one part of physiology, and Derone has another part of physiology, but she realizes that they have a lot to do with each other. And in fact, they fit. Okay? There's part of the model that Derone presents that's also part of Jane's model. But then they diverge elsewhere, right? So they've got this point of contact, but they're modeling different things. And gosh, she says, if I could put the 2 models together, it would be a larger, more comprehensive model. It would be more faithful to the real underlying physiology, and we would better understand what's going on, either to understand the pathology for drug design or for patient-specific modeling. Okay? So that's what she wants to do, but how do you do it? Okay? Well, Derone has a publication, of course, so maybe she could read the paper and just rebuild it. Unlikely. Okay? It's not going to work. First of all, it's very time-intensive. But secondly, well, publications themselves typically don't have enough detail for replicating the model, right? This is the reproducibility thing. And it's not really Derone's fault. The publication has a page limit, or these details are just too small and picayune to put in there. Now, if it's a good journal, or should I say a modern journal that understands reproducibility, then maybe it'll have a supplementary section that'll have some of the details. But experience has shown that that's rare, and that even when that's there, it's not enough. Okay, so it's not going to work. So if Derone is really good, not only is there supplementary material, but maybe he's actually deposited the entire code on GitHub, and it actually works. So this is unlikely, but it would be nifty if it did, The problem is that even if it runs, she needs to connect it, right? So if there's a model on GitHub, okay, great. Amazingly, it's actually reproducible. She can take down the model and she can produce it, and it does the right-hand side. The problem is she needs to connect it to her model. So she needs to understand the model, which variables in Daron's model are what things to connect to her model. And if Daron just called things X, Y, and Z, that's a problem. Okay, so that's not going to work either. Really, the only solution is to hire or steal Daron's postdoc. Okay. So this is successful, but I hope I've convinced you that there's a problem here. Okay. So as I stated, the publication itself doesn't have enough information to be reproducible. If by some fluke, very unusual scenario, the author has deposited the code and it is reproducible, it's still not good enough because it's not understandable. She can't connect things together. So to fix this, I argue we need standards and annotation. Okay. And I'll give a description exactly what kinds of things we need to annotate. We need to annotate the entities in the models. And in particular, it's really important to annotate those things that are going to be the points of intersection. Of course, you don't know a priori where the points of intersection are, so we tend to annotate most things in a model. Okay. Now, I want to emphasize that when we talk through what SemGen does, and I want to be clear that SemGen, our tool set for model merging, is not fully automatic. This process of putting Lego blocks together is inherently imaginative, right? That's the human endeavor. We're never going to get a computer to figure out how to put models together. That's up to humans. But once a human has the insight, then the computer should make it as easy as possible. And that's what we strive to do with SemGen. Okay, now I'm going to switch gears to, so that was sort of at the abstract level with just little boxologies of models. I'm going to give you a real example with some real models that were published that we merged together using our tools. So this is going to be in the realm of vascular smooth muscles. And again, if you type into Google search or YouTube, calcium and vascular smooth muscle contraction, you will get 1 million hits. You'd think something like that is pretty specific, but no. So the vascular smooth muscle surrounds arterioles, and they, like any muscle, can constrict. And the whole point of them constricting is to control your blood pressure, right? And so as they constrict, the blood pressure goes up. As they relax, the blood pressure goes down, and this allows oxygen to get to other parts of the body that might need it more. And so, like I say, you get 1 billion pictures of this process. And here's one that I picked at random. And I picked it because it's relatively simple and because it shows calcium, okay? Because that's going to be the focus of my example is calcium. So calcium comes in, and basically, if calcium comes in, if there's more calcium inside the cell, then you get contraction. If there's less calcium and more nitric oxide, then you get relaxation. Okay? All right, now here's another picture of the same process, and it's actually enlightening to see how many different pictures there are. And this is one of these problems where standardization needs to happen, right? How the heck is a computer going to understand that this picture has anything to do with the previous picture? It's not, okay? So this is, believe it or not, the same thing. You can see a few of the players that are the same thing. There's the calcium channels up there, and down here, the little red circles are all calciums. And I want to highlight, this is a published model by Capella in 2008, and this is one of those rare publications that actually has source code with it. And the source code was deposited in the CellML repository. Okay? You can look it up. And I wanted to focus on one aspect of this model, IP3 here. I don't remember what IP3 stands for, but it's some receptor that opens and shuts the gate, okay? The calcium gate. And this time, this is the SR, not the membrane, not the cellular membrane. I don't even know if it works on the extracellular membrane. But at any rate, it is a gating receptor that opens and closes these ion channels, which obviously affect directly the concentration of calcium. And again, that concentration of calcium is what's going to affect the cell contracting. Okay, now in the Capella model is big. It's got lots of players, lots of different things going on. It's not just about calcium. All kinds of things are happening. It's a broad model of what's going on in these cells. And so when it looks at how to model IP3, it uses a very simplistic mathematical model of how IP3 binds to and opens and shuts that gate. Turns out we understand a lot more about gating than is encoded in this model. So there's another publication that actually came before by a fellow named Snade, who has a multi-state, more sophisticated, more complex, more interesting model of how IP3 gates the calcium ion, the ion channel. And this is a different kind of schematic, but the capital letters, O is open, S is shut, A is active, I is inactive, and R is the receptor. That's where IP3 comes in. And again, here's a publication where the mathematics are actually encoded and provided as source code that can be run, written in SBML, in biomodels. Okay? So here's a whole nother problem I haven't talked about, is that people use different languages for encoding the processes, right? They don't use the same mathematical modeling language. They use a variety of different languages. So what we want to do is we want to put these two things together, right? Just like Jane. We want to put together the Capella model with the Snade model, so we get the breadth of the Capella model, but we have a sophisticated, interesting model of IP3 gating of that ion channel that Snade provides, okay? But we got the source code, but one's in SellML and one's in SBML. So that's another problem. And as you might guess that I'm giving these examples, this is a problem that we can solve. Okay, so that's one of the things that the SemGen does. The SemGen can read biosimulation models in either of these languages and unify them together. And so that's what SemGen is going to allow you to do. As I said, once you have this idea of how to put the models together, then the tools should make it as easy as possible to do that. So this is what SemGen can do. And I guess the first slide I have is a schematic of how that works, which is that you need to annotate the models. As I said, you need to know what the variables mean. You can't just use letters for particular things. So the two models on the left are in different languages. So again, the tool can simply read those languages and put it into a common format we call SemSem. And then you annotate the model, so now we know the semantics of all the variables in a consistent fashion. Oh, darn it, it's auto-advancing. OK, so that's the first step. Step one is to annotate and create SemSem models. Step two is to actually do the merge. I'm not going to say too much about this, but basically it's going to notice those similar semantics and prompt the user, do you want to connect these two things? And once you connect the two variables, then you also have to decide, well, which mathematics are you keeping? Keeping the Capella or are you keeping the Snade? Okay? That's the process number two. And finally, number three, that's all within SemGen. Number three is export, right? And so we want to output to SellML or SBML so that the execution can actually be run. So SemGen does not do the biosimulations executions at all. Okay, that's a separate process. But we use other people's software to do that part, and it works just fine. Okay, so then here's a screenshot, and what you're seeing is a visualization of the two models, or the two gray lumps. The bigger model is Capella, and the smaller model is Snade, or Sneed. I don't know how you pronounce the poor guy's name. And I'll just point out that there's a variety of different ways of visualizing these models. This is using D3 technology, so you can do all the kind of network visualization stuff people are used to these days. Yeah, and that's all I'll say. And then I'll zoom in. So here are the two models. And this is post-merge. When you're doing the merge, there's a whole other graphical user interface that shows up that says, well, are you interested in, and then here are the 4 points of contact. Are you interested in the probability of IP3 being opening the gate? See, and that's that third one down. And then the bottom one is the calcium concentration. And then there's the volume of the compartment and IP3 itself, the concentration of IP3. So those are the 4 points of contact that you have to link to put the two models together. And as I said, for each one, you have to decide which mathematics you're going to use. And you're going to choose the complicated mathematics, and that's how you do it. OK, how am I doing on time? Good. OK, how do we make those connections? So I'm not going to spend time today going through the ontology of physics for biology. But really that's the answer, is that we can annotate in a uniform manner things like concentration of calcium ion. And we can annotate that we're talking about calcium 2 ions per Kebi identifier number blah, 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 right? So we use the ontologies to say that, yes, I'm really talking about the same physical entity, calcium ions, and I'm talking about concentration. Same thing with a particular gate and IP3 and all those participants and all the reactions and processes that might be happening. We use a common language for annotating them. Okay? Great. Yeah, so I think I'm going to have a little extra time for questions. So that's my model merging example, and I'm going to do a little summation sort of SemGen. So as I said, you annotate against standard ontologies for physical entities and those entities' properties, okay? Physical properties of things like concentration of, if you're a fluid model, it might have fluid flow, masses, charges, voltages, those kinds of properties. And then we can visualize models. I'll show you one visualization, but there are several others possible. We can merge models or extract models, right? So another thing you might want to do is throw away part of a model and say, no, I want a simplified version first before plugging it into mine. And then once you annotate these models, then you can do more semantic searches. Give me all the models that mention calcium ions, for example, is something you can do. And then again, you automatically notice when two models that you might be merging have the same kind of entity in them. And so that's what the match point notification is. And so we now have a pretty stable and well-tested piece of software. And I'll talk more about how that happened in a minute. And we do the annotation, as I said. Here are three different descriptions of different visualizations. You can have sort of the dependency view, which I believe is what I showed. And that view simply goes through all of the equations and says, well, what things participate in what equations? And that's the mathematical dependencies. You might also have a hierarchical view. And then I'm not sure whether the picture was physium or not. Or then you could have a process-oriented view, where every link means that there's actually a process among those two participants. And then, as I said, there's a model merging interface, which I'm not going to show. OK? And of course, since we're trying to be reproducible, and since this is NLM-funded work, you can go and retrieve this software and try it yourself. OK? So to wrap up with SemGen part, we've got annotated models. We can do semantic search. We can merge models. Yay, we can take off, cross the blue sky. Maybe not. So I'm going to talk a little bit about some of the problems. There are certainly problems. And these are some of them. Actually, the first one is pretty amazing. I'm excited about that. We're currently drafting up a manuscript on this result. What we did, well, first of all, what happened was there's just not that many. I mean, so at some level, the model repositories are big. So the PMR, both the CellML repository and the Biomodels repository have hundreds of models, probably close to 1,000. So that sounds like a lot, but a lot of the models are kind of about the same area of physiology. So there's a lot more that's not there. So we started realizing, well, shoot, there's no model for this. What's going on? Or why is this not here? There's lots and lots of publications about models, about, mathematical abstractions that predict something that actually has code even maybe and runs and produces a simulation. There are lots of papers like that. But there's not many of those papers where the code actually gets into CellML or into biomodels. It's embedded in Fortran or in C++ or in some site-specific code, actually, that doesn't run anywhere else. So what we did is we're doing sort of a systematic literature review, OK? So we type in some search words, and this is preliminary work, so I can't give you the details yet. We type in some search words and retrieve a whole bunch of models, a whole bunch of papers that might be biosimulation model papers. So 6,900 retrieved articles. That's too many to go through. So we do a randomization of them, and we select randomly 150 of them. Of those 150, some of them the full text is not available. We want to look at the full text because the code might be buried in the full text. But that's really not the problem. These days, most are available in full text. Many of them aren't really, our search terms weren't good, right? Of the 150, only 44 were actually about biosimulation models, and their full text was available. But of those 44, exactly 0 had source code. I mean, they didn't even have a sentence at the end of the paper, for source code, please contact the author. There was nothing, nada. There was just some equations and a statement that claimed something ran. In many cases, it actually even said the language in which it ran. But there was no indication that you could get the source code. Never mind having it in a supplemental material. I mean, I'm hoping that as we expand this number of 150 to, say, 350 or something, that we'll find at least some papers that will have some supplemental material with more information. But right now, it's looking pretty grim. And it does make sense based on our experience over the last couple of years. Gosh, we just can't get the models. Well, they're just not out there. People haven't yet gotten on the ball about this. I think the tide is changing, but that's what we found so far. Annotation is expensive. I'm not going to talk about that. But then the other thing we know from talking to CellML and SBML curators is, oh my God, this is a problem. Okay, it's kind of always the case that when a paper gets submitted and someone says, I want to publish my model in SBML, and here's the source code. Okay, here's the SBML code, and gives it to SBML. The SBML has a team of curators that goes through these models one by one and tries to replicate what's in the paper. And what I've heard is that 9 times out of 10, it doesn't replicate. I go back to the author and say, hey, it doesn't replicate. Oh, yeah, you got to set this parameter to that. Oh, yeah, I forgot this bit of code. Oh, it's always an iterative loop. And so that's the curation process. And eventually it works, but there's that step that seems to be missing many of the times. And then in CellML, we actually know that for whatever reason, and it could be because the types of models are different between the two domains, between the two libraries. The curators have many examples where they tried to implement it and simply failed. It was not reproducible. And that's all there is to say about that. So yeah, so those are some of the problems. Along with the cost of annotation is the third problem that I haven't talked about, but that's kind of obvious. It's expensive to do that. So what are our responses? Responses are you need to build infrastructure, you need to build a community, you need to build a culture, and you need to emphasize reproducibility. Because those are the two things that solve those problems, I think. So we've been pretty successful building a community and building infrastructure. This is one of the sort of, it's kind of hard to write about, but in my mind, I got to work on this. It's one of the most exciting results so far, is that we've really been able to connect, in most cases, through this community called Combine, Computational Modeling and Biology Network. They have basically 2 meetings a year. So we've gone to all of those, or most of those, and presented our work. And people have said, yeah, I'll sign up for that. Okay? Because we've been pretty successful in getting them on board. And so, in fact, recently, 6 months ago, or less than that, we created a whole consensus document where we got all of us, 24 co-authors, to write a paper together about what the right way to do semantic annotations are and what the benefits are of that. And so that has been a big success. And it's not just, it's not just the 2 meetings a year. It's actually been more than that because there are other meetings like cardiac modeling, and there are site-specific modeling meetings for heart disease or for diabetes, and we've gone to some of those. And again, our feeling is that people are coming around to this notion that we need to be more careful about the semantics of our models so that people can reuse them. Okay, and then reproducibility. And so our response there has been actually to write a whole grant proposal around this notion of reproducible biomedical models and say that, wow, this is such a big problem. we need a whole pile more funding. And so we just recently received funding for this, June 1st, 4 days ago, 5 days ago. So I can't tell you much about it. Our kickoff meeting is the 25th. It's a multi-site grant. It should be a 5-year grant. The point is, well, so some of the points are slightly different from what I said. One of the points is that you actually ought to encourage reproducible model construction from the get-go as you're building the model. Never mind once you're ready to publish it. You need to do it as you're building it. And so that's one of the big goals of this center. And then, as I've talked about today, model annotation, search, and model composition. That's the merging task. And then the third part is, as I alluded to in my merge story, we just hand it off to a third party and say, OK, go execute it. But really, you want to visualize. The visualizations I've shown you in SemGen are just static. Those are the players in the model. Those are the processes in the model. But you want to actually see the model run, well, then you have to use a different software. So this center is going to say, no, we're going to make it all unified so that you can search for models, you can visualize them the way SemGen does, and then you can also run them and visualize those executions and try a variety of different execution engines. And so that's the third goal. And so the center should it's going to have funding for curation. It's going to have funding for annotation. So we'll try to offload some of that. It's not up there, but one of the exciting things is that we have a number of journal editors who wrote letters of support for this. So the notion is that in addition to trying to get a culture of annotation reproducibility, if we get the journals to say, hey, if you're writing a paper about a model, you better give us supplemental material with the code or whatnot. And so that's part of this grant as well. And then, as I said, access to actually running the models in a biosimulation environment, or what kind of assumptions you're making. There's different choices of mathematical paradigms. Data annotation, you want to annotate the data with which you use either to build the model or run the model, and then visualizing the results of those. So that's the Center for Reproducibility. I just want to end by teasing apart a little bit the difference between reproducibility, as this center is promoting it, and reuse, as I've talked about it today. So I think, as the story as Jane and Daron told, simply reproducing results is not enough. You can download someone's code, and yes, it reproduces what they claim in the paper. That's good. That's important. But it's really only a first step for this more imaginative process of model recombination, okay, and reuse. Because you don't want to reuse something exactly as is. Instead, you want to tweak it. You want to plug it into yours. You want to change it a little bit. You want to explore it. And so you need to be reproducible, but you also need to have this shared semantics of what the model is doing, what its assumptions are, what its mathematics is, and be able to visualize that. So then you can put the models together as you see fit. All right? So to conclude, I think modeling physiology is hard. It's about processes that unfold under time at different scales. That's kind of what makes it hard. Whereas modeling, the genome or modeling static things is very different, and I would argue simpler. And then to really do this sharing and rebuilding from others, it requires common standards. Okay, standards that are ontologies for the processes and the properties and the entities, and a community of people that are willing to invest in the time and effort it takes to annotate them. And I think reproducibility is great, but I view it as a step towards model modularity and reuse. That's my talk. Okay. I just want to end by emphasizing these 5 people. They've really been very tightly involved. It's been a joint grant between University of Michigan and ourselves. Brian Carlson is from Michigan. He's done a lot of work in understanding the physiological system. He works in hypertension. Dan Cook is the author of the Ontology of Physics for Biology. Graham Kim is my grad student who's doing this systematic review. Max Neal is the genius and originator of SemGen, the software itself, and Chris Thompson is the programmer. So thank you again, and I'll take questions. Yes?
So about SemGen itself, is it a together and identify where they're connectable, as well as a repository of models?
It is just the former. It is not a repository. There's no repository there at all. So it pulls from the CellML or SBML. We were not going to reduplicate their work.
And you have explicit connections with those two?
Yes. Yes. And JSIM as well. But that's one of the things the Center for Reproducibility might explore, is if we can expand the set beyond those two languages.
So do your findability capabilities let people go to one place and find models in both CellML and SBML?
That's the idea. I think the last time I talked to Max, he said this still wasn't working. We can search over, once we've annotated and made things SemSim model, we can search over them easily, but we can't right now search over them. That's definitely something that's the vision. You've got it right. That's what we want to do, and definitely that's what we will do at some point. But I don't think I can promise that today. Yeah. Which? Yes. I mean, the answer is yes. Right. Right. Yeah. So SemGen is publicly available right now. And right now, with this R01, SemGen was created as a software package that you have to download and install. We've learned that that's actually a barrier these days. And so one of the things we'll probably be shifting to is a more web-based platform for doing this. Is that what you were getting at? Yeah, so right now you would have to download it and install it. But yeah, if you had a model that was private that you were about to publish that was written in CellML or SBML, you could download Protégé, pull in your model, and browse other models and see what plugs together. Right. So to date, they've been mostly informaticians, right? And that's kind of the model, is that you don't want the biologist to have to learn all of these ontologies and all the proper knowledge representation and all the proper semantics. You try to make it as intuitive as possible, but the expectation is the biologist is going to submit a model, and a curator and annotator will go through it and annotate it. Now, Brian Carlson is both, right? He knows, and Max, both of them are both. They both know the informatics and the biosimulation modeling, the physiology. So it's an open experiment to what extent we can train those people and use it. We did actually test our system with graduate students and postdocs who had completed training in physiological modeling, not in informatics, to see if they could use it. And it worked pretty well. So it's kind of an open question as to how easy it really is. It's hard to measure that kind of thing. Right. Yes, exactly. Mm-hmm.
But the problem is that most of the experimental biologists do not have any idea or do not understand the standard at all to use it.
Right. So experimental biologists, wet bench folks, are different from mathematical modelers, who are different from informaticians who know the knowledge representation. So it's an ugly problem. Yes. We've mostly been working with the mathematicians. That's the other set of meetings we've been going to, is mathematical modeling meetings.
So another chasm is between research and clinical practice.
Yeah, right.
So this is for bioresearchers and co-researchers. But what you're modeling at the end of the day is disease.
Yes.
And data are going to be EHRs and things like this. So how do you see the possible connections?
Right. We do. We have a paper submitted using FHIR and stuff using not FHIR. Oh yeah, I know it's using FHIR. To get clinical data to try to validate the model. So one of the interesting issues is, and we throw this whole story since we have a whole paper about it, it's under review, is that currently if you have a mathematical model, you're a mathematician, you create a mathematical model, it seems plausible when you run it, but you want to validate it before you publish it. So you connect up with a wet bench experimentalist who has a mouse, a physical mouse model, a critter, and you measure, or tissue samples, or whatever it might be, and you actually measure tension in the muscle, right? You can do that, and probe it and see if the actual physical cell behaves in the way that the model predicts. So you do the experiment afterwards. Needless to say, it's very expensive to proceed that way, right? Wet bench work is always expensive. So instead of doing that, I think what you're getting at is why not just mine the EHR data to see if the EHR data matches the model. There are a lot of problems with that. And the problem is the clinical data is typically measured at a tremendously gross level for what we need to do. So the experiment we ran is right heart catheterization procedures, where you actually stick a catheter into the heart, and then you're inside the heart. Now you can measure pressures in different compartments and resistances in different vessels and all that kind of stuff that you typically can't measure. So not everybody gets right heart catheterizations, but heart transplant patients do. And so we got a whole bunch of heart transplant patients. We look at their data, and we're trying to match a model against that. So that's one of the papers we have under review, is to look at that. It's going to be a ways before that really plays out, I think. Yeah, in the back. Right. Yeah, well, this is your question as well, and it's kind of the jury is out on how much we can do there. You know, I really should have had SemGen up and running on my machine, because then I could just show you. I don't know if I dare. I don't think I dare do that. It currently does try to do a lot by itself, right? So if you've got a mathematical model coming in, one of the things that happens is, in some languages at least, those elements have unit information. And it turns out from unit information, you know, micromoles per unit area or something, unit volume, you can deduce, aha, this must be a concentration, right? And so the tool, when it reads one of these models with units, automatically does a fair bit of annotation. It already classifies variables as different types according to the units. So we do as much of the work as possible. That's the whole point. Whether or not we can set down someone who has no familiarity with this kind of stuff and walk them through it, that's a little trickier. One of the things that we find happens is that experimentalists have all these shorthands, all these simplifying assumptions that they use to talk about things, that when you break it down, the physics into the details become a lot more complicated. One of the physics examples from the OPB that I like to tell is voltage. So you could either talk about the actual voltage in, I don't know, micro whatevers, between the potential energy between inside and outside the membrane, or you could count the number of calcium ions. It's the same, it's getting at the same thing, but the physics involved is fairly different, right? So that's an example where there's this, that's why we drop back to the physics, to the ontology of physics for biology, in order to clean up and clear up those semantic differences. Did that help? Okay. Other questions? $2 o'clock? I mean, is it 3 o'clock? 10 now still. One more. It's a bit early. Go ahead.
So going back to the divide, you know, there's the same language on the research
Right.
side and the clinical side. So how do you plan to purchase? Because this, I mean, what is it? The ontology.
Physics for biology.
For biology is not the ontology of physics for healthcare. So
Right.
how do you plan to bridge the divide?
Well, so again, it's all physics eventually. So that's why we look specifically at things like pressure, right? So we're not going to use large swaths of the clinical record at all. It's only numeric values of things like pressures and masses and rates of change, liters of blood loss, things like that that are quantifiable and are sort of physics-based, that are going to make sense in that framework. All the subjective stuff is pretty hard to put into a model. So I think it's only a tiny portion of the clinical record that will be useful in this manner. Yeah. Uh-huh. Yeah. Uh-huh. So that's an automated process. Again, it has to do with this physics view of the world, right? So at the end of the day, you're talking about some physical entities, chemicals, molecules, proteins, physical things that are interacting with other physical things in a process that occurs over time. That's a reaction. And so it doesn't matter whether the code is written in CellML or in SBML. We can parse that and figure out that things they're physical entities and biochemical processes or physical properties. And then SemGen is just the knowledge representational notation we use for storing all that information. We can output that in OWL or RDF. But yeah, that's just an internal notation. And then after we merge things, we output them in CellML or SBML. There's some interesting questions around loss of information when those translations. But I'll go ahead.
I was wondering, so basically the whole purpose of this model, or it's basically like a predictive model, you get it
Right.
and then you extrapolate it to predict something else.
Yeah.
Has not been proven experimentally. So I just wondered, in your experience, how much of those extrapolations have actually
You're asking what are the success stories for the whole field of biosimulation modeling. Yeah, I'd like to say, oh, well, we realized that this process occurred and that if we intercepted a drug right here, it would interrupt it and cure X or cause Z or something. That's a good question. I should have a portfolio of success stories, but I don't.
Have you seen any?
What?
Have you seen any?
So again, this is not my field, right? So the right person to ask would be Brian Carlson, and I'm sure he would give you lots of success stories. They do a lot of modeling of hypertension, of kidney disease, and cardiac. Those are the three wires. There's a most published is in cardiac modeling. So there's a whole range of interesting models around the shape and the 3-dimensional shape of the heart, how that changes over time, and the voltages and all that. So yeah, I'm just not the right person to answer the question. Yeah, in the back. Thank you. It all began as cardiac simulation, yeah. Yes, I certainly can. Right. Right, so I think a lot of this is around hypothesis generation, right? So yeah, and so Olivia gave one example of clinical data, but to my mind, the better example would actually be repositories of those experimental data that were designed for physiology. So if someone measured tension in a muscle cell for researcher X for model Z, that data should be available for a different researcher with a different mathematical model. And to use it. And so that's definitely one of the things that the Center for Reproducibility is aiming to do. No, there are not data repositories right now. That's what we're going to build with the Center for Reproducibility. Well, both, but the model repositories already exist to some degree in the SBML and SLML repositories. But yeah, so connecting it to data is really, really important. The other connection to data science, though, is all about the reproducibility and annotation. I mean, that's part of good data scientists, right? So just as I'm arguing that you need to model what your biosimulation model is about or what it does, whenever you collect data, you need to annotate and talk about exactly what that data is, how you collected it, under what circumstances, what temperature, what atmospheric pressure, all of that stuff needs to be captured in a mineable, reusable manner. And that's definitely part of the Center for Reproducibility in this new grant. That's right. That's absolutely correct. I mean, you know, yeah. So I think 10 years ago when I started in this direction, one of my insights was from software engineering, right? Computer programming, that's my background decades ago. And so that's what they tell you there. You got to start documenting your code from the start, not post hoc. And the same lesson applies here, exactly. And so that's the connection to data science, I think. Emulate what?
Test during development. You write the test first, then you write the code supporting the test.
Yeah. So that's kind of the software engineering idea. Okay? Yeah. Use, yeah. I don't think we've gotten there. It's an interesting point. Right. Yeah. Yeah, well, so right now we have so few cases of the kind of model merging to talk about that we wouldn't be able to measure that. It's just the last question. Go.
Has somebody taken a merged model and put in biologically plausible input data, a full range, and produced test data from that?
Yeah.
Is that a publishable paper? Is that
So-
a unit of information that can be used to reward people, so to speak?
Yeah. So that's some of our earlier publications, actually. Actually, the trajectory picture I showed was from a publication of ours a long time ago that actually was from a merged picture. I mean, it was just easy to grab. That's why I used it. So this picture here. Yeah, so PHT. PHT up there stands for Pandit Hinch Niederer, Pandit Hinch Tran. 3 different papers that we merged together for cardiac, for baroreceptor, I can't remember what modeling, but it was a while ago. And so we published it and we showed that you could put them together and generate plausible results. Now, we didn't take the next step and go back to a mouse model or whatnot and see if it was validated, but other people had done models of mice and seen this kind of pattern. And so we matched these against that. And so we actually reported the, I don't know, this chi-square difference or some kind of measure of how well our curves match to their curves. So we've done that work. Is it common? Oh, you know, the other thing I could point to, do I have this backup slide? Not in this version. There's a paper done by the EBI folks that looks at all of the models for beta cells for diabetes research. And they did this tremendous job where they searched the literature. And I think there are about 30 papers. And they put together a diagram that connects the models. But the way they did it is they read the papers. And if paper A said, oh, our model is based on something built by paper B, then they drew a line between the two of them. So what we want to do now is actually go and collect all those models and see if we can plug them together. So that's another example of where it could be done, but hasn't been done yet. Okay, now are we out of time? You guys came up with a whole bunch of good questions. That was great. Thank you.
Thank you, brother.
Yeah.